Hello, everybody. Then I will continue presenting some of the work we did in task 1.3, where we looked into the dissolution and solubility and reactivity of the test materials in different biologically relevant media that we applied in the Patrols project. Concerning the media, we looked into cell media, we looked into the long-lining fluid, the fecal lysosomal fluids, as well as the gastrointestinal fluids. And we looked into determination of the solubility limits at different time points, looked into determination of the dissolution rates, pH reactivity or causticity, redox potential, and the ability to form reactive oxygen species. Some work was also set to look into phase transformation and reprecipitation, but that will not be discussed in this presentation. We used four main methods for the solubility limit and dissolution rate determinations. We used a so-called sensor-dis reader method for testing in vitro media. Here we tested 8 different cell incubation media. We tested all nanomaterials aligned with the tox work packages, and all nanomaterials were tested in 3 different media. We developed an atmosphere temperature and pH controlled stirred batch reactor system in which we tested the solubility and reactivity as well as the dissolution rates in gambles and fecal lysosomal fluids. And here we tested all nanomaterials, and one experiment was also conducted at ETU as part of the PhD project with Elsa Holmfrid out there, looking into the dissolution of cerium oxide. At BASF, they used a flow-through cell membrane for testing the reactivity, kinetics of dissolution in gambles and fecal lysosomal fluids, and a sequential batch reactor dissolution set up for gastric solutions, and in both cases they tested all nanomaterials except from the carbon nanotubes. Reactivity was studied using different methods. Here we used the sensor-dis reader together with the dissolution studies to determine the variation in oxygen concentration as a proxy for redox behavior and pH in the sensor-dis reader plates. And we measured the redox potential at fixed pH in the stirred batch reactor system as an indicator of reactivity in the media. And then we used the FRAS assay to determine the ferric reduction capacity of the particles in serum. We used the DCFH2 probe in phosphate buffered cell line, and then we looked into the electron parametric resonance spectrometry for specific reactive oxygen species. We also used voltammetry to look at the specific redox potentials of the nanomaterials in different media. But let me first jump directly into the experiments we conducted with cell media. Here we tested 8 different media lined up here at the top of the slide. We tested the dissolution or solubility time points, the endpoint solubility at 24 hours in the cell incubation media. And particles were incubated in these cells where we had test material on one side, and then the other side was left as a control. And in each of these wells, there's a small sensor for pH or oxygen concentration measurements, which we then can measure as a function of time. And all experiments here were conducted under in vitro cell incubation conditions, so the system was placed in an incubator and basically trying to mimic as much as possible what is going on in the in vitro assays. So looking here at the results, we see that the amorphous silica materials are highly dissolved in the test media investigated here. We see that in all cases we have the highest amount of dissolved material for these amorphous silica materials, and then the second highest is the zinc oxides, where they are most dissolved in medium 8, which is the LHC medium, which is used for bronchial epithelial cells. We see that the silver particles have a relatively low solubility, barely detectable, and also serum oxide has a really low solubility. We see that DQ12 dissolves to some extent, and this might be due to an amorphous rend on some of the quartz particles, as we've seen previously and reported in a paper by Rosgaard et al. So this could explain that this otherwise crystalline silica material is somewhat dissolved also in cell media. We see that the NM220, the barium sulfate, is more or less equally dissolved in all cell incubation media. If we look into the carbon nanotubes, we see that very low amounts of metals are dissolved from the carbon nanotubes, and this is of course also due to the start concentrations in the doses given. We do use a fixed dose of 0.32 milligrams per milliliter in these experiments, so this is a relatively high dose, of course, compared to the in vitro studies, but it is a dose where we can clearly measure the dissolution and reactivity. We see that the highest values is for aluminum in NM400, where we have about 0.3 milligrams per liter that can be dissolved over 24 hours in these experiments. And this was in medium 5. Then I think I will jump to the reactivity test just to take a little bit of a look here. Now we're already talking about the sensor dish reader method. And in fact, here we measure the delta pH, or show the delta pH and the delta oxygen concentration, which is then the difference between the wells with particles and the wells used as reference. And we see that there's a somewhat different behavior of the particles in the same media. So they don't follow the same reaction path in the different media. Here, for example, we see a relatively strong initial reactivity of NM200 in regards to changes to pH. The pH scale here is from -0.6 to +0.6. In most cases, we see a little bit of a reduction in the pH, and then sometimes levels off or even becomes positive after or during time in the experiments. Some materials cause a increase in pH, for example, what is seen here for the silver from Sigma and the REF silica in medium 5. We see that titanium dioxide and zinc oxides typically increase the pH, and that has also been observed in other studies before. When it comes to the oxidative behavior, we see that relatively moderate reactivity in all cases, but most pronounced in LHC, medium 8, where there's a clear initial reactions for most of the materials in the first few hours of the experiment. So on top of showing different reactivities of the different materials in different media, we see different reactivity of the materials in the media, and also that we have a clear kinetic effect of reactivity. So in most cases, reactivity happens within the first hours of the incubation. If we now take a look at the test done in physiological simulant fluids, we looked into the Gamble solution, faecal-lysosomal fluids, and the gastrointestinal fluids, where we tested all materials in Gamble's and faecal-lysosomal fluids, so pH 7 and pH 4.5. And in the sequential gastrointestinal fluids, we did not analyze the carbon nanotubes. There was also a request for analysis of dissolution in blood and plasma, but we did not have that method in place, and I think we can use the results in Gamble solution as a proxy for the behavior in blood and plasma. But of course, a verification of that would be nice in the future. Looking into the results, we see here for the materials in faecal-lysosomal fluids and in Gamble solution, the time point solubility limits in the reactors for 0, 1, 2, 4, and 24 hours, and then we see the logarithmic conversion here at 24-hour concentrations. And clearly, the zinc oxides are heavily dissolved in faecal-lysosomal fluid. Basically, everything dissolves almost instantly in faecal-lysosomal fluids. We see also that barium sulfide is dissolved to some extent. Low values are seen for most of the other materials, in particular for cerium oxides. DQ12, we see some moderate values, but still a continuous dissolution, as just discussed before also. We see moderate dissolution of the amorphous silica materials and a very clear higher level of dissolution in the Gamble solution, as shown down here. So clearly, the amorphous silica reacts a little bit different than the other materials. We also see that barium sulfide is dissolved to some extent, to some higher extent here more or the same extent in the Gamble solution. Zinc oxides are dissolved, but to a lower extent than in faecal-lysosomal fluid. Titanium dioxide we do not see to dissolve. If we look into the dissolution rates, we have the flow-through cell here, and we see more or less the same picture here with the faecal-lysosomal fluids and the Gamble solution down here, and high dissolution rates for the zinc oxides, where we find the highest rate for NM110. We also have a significant dissolution rate for the silver from sigma and barium sulfate we see here. And when it comes to the Gamble solution, the highest solubility dissolution rates are found for the silica materials and also DQ12, probably related to the amorphous layer, as we mentioned before. We still see a significant dissolution of a rate of the sigma silver, whereas the rates for the zinc oxides is relatively moderate in the Gamble solution. If we compare now with the data for the batch reactor system, we see that there is some similarity between the results using the two different methods. And here we just have the 24-hour concentrations in the batch reactor system compared to the rates. And if we try to make a correlation between the two, we see for the Gamble solution that we have two different linear trends. One, I would say, for the regular behavior over here on log scale for the dissolution rate and solubility limit at 24 hours, where the two silver materials and DQ12 plot on their own line, where we see high rates but not so high solubilities after 24 hours, and that's probably related to the limiting factors of solubility when it comes to the silver and limiting factor of amorphous material being removed, and then you are left over with the crystalline core of DQ12 in the end. At least this is my interpretation at this point in time. When we look into the fecal lysosomal fluid, we see a more coherent picture with a decent linear relationship between the rates and the solubility limits at 24 hours. This is, of course, good to see, and we also see that probably there are less rate-limiting effects in the acidic fecal lysosomal fluids. If we now jump over to the dissolution in gastrointestinal media, we have tests where the particle spent 5 minutes in saliva simulants and then moves into the stomach for 120 minutes, and then we conclude with 120 minutes in the intestine. Here we see zinc is dissolved already, and barium sulfate, and also the silica materials to a relatively high amount already during the first 5 minutes in saliva, and this continues then into the stomach where it's acidic conditions and the zinc oxides dissolve even more. We see relatively stable concentrations of barium sulfate and the amorphous silica materials here, and then we move into the more basic intestine where we see the solubility of zinc oxides dropping, so we have a reprecipitation occurring, whereas barium sulfate and the silicas are dissolved a little bit more when it comes to the silicas, but staying more or less constant for the barium sulfate. So it's important to note these differences, and also that we have this more or less similar behavior of the barium sulfate and silica materials in all compartments. Then I will jump over to the comparative reactivity testing we did in bracket standard methods. Here we applied also 3 control particles, so carbon black Printex 90 and manganese oxide and copper oxide, known to be oxidative reactive materials, to have some positive controls, as the materials in patrols are not that reactive. We already looked into the reactivity in medium 5, so the DMEM, and we chose DMEM as our medium for testing with the sensor reader method for also these materials. We already documented that we see different pH reaction behaviors and different delta oxygen reaction behaviors. And if we now look at the materials that we studied here in the comparative testing, we do not see a significant higher reactivity of the manganese oxide or copper oxide, maybe, or not for the, carbon black either when it comes to the pH. When it comes to the delta O2 values, we see that the Printex 90 is the most, is the particle decreasing the delta O2 most, but the oxidative behavior of the manganese and copper oxides cannot really be resolved in the DMEM experiment. What we do see, however, is that the reactivity is happening within the first hour of the experiments for all particles. So we see that we start at very low changes, and then after 1 hour we already have more or less the changes that we would see for the different particles. So I'll shrink now the data for the SDR in DMEM to just the 24-hour and minimum and maximum values, and then compare with the cyclic voltammetry data. So cyclic voltammetry is measuring specifically the redox behavior. So blue is reductive and yellow is, red is oxidative. And here we see also that in DMEM, in fact, it's probably true what we see in the cell medium that for the sensor disk reader method that the reactivity is quite moderate. However, in this study, they found that ISTEC some reductive behavior of the NM105 and also of the NM110 zinc oxide and the small oxidative behavior of the sigma silver from sigma. When we move into the more standardized test media, we do see the positive controls pop out now. Still, we do see some reactivity of the zinc oxides and silver and also serum oxide. In Gamble solution and fecal lysosomal, we see somewhat similar trend, but we cannot see a reactivity of Printex 90 in the fecal lysosomal fluid. If we compare that with the redox potential we saw in the batch reactor system, we now have our particles in fecal lysosomal fluid and Gamble solution alternating down here for each of the particles. What is important to note here is that we see overall a similar behavior of the particles in the different for the different particles in the two different media. However, we do see typically stronger reactivity in the fecal lysosomal fluid. So we see it here for Printex 90, and we see it for manganese oxide here and for copper oxide as well. We also see that the zinc oxides to some extent, but in particular the silver from sigma is also quite reactive in both fecal lysosomal fluid and Gamble solution. For the silver oxide, for the silver particles, we actually see both oxidative and reductive behavior, depending on at what time we look at it in the experiment. Serum oxide also comes out as relatively reactive, in particular in fecal lysosomal fluids. If we now compare with the FRAS assay, we see that the Printex 90 and manganese and copper oxides are significantly reactive, as well as the zinc oxide NM110 and the silver sigma. The other materials are more moderate in their reactivity. If we look into the electron paramagnetic resonance spectrometry, we see that if the test medium or test probe is CPH, then we do see that the positive controls clearly step out. The carbon black and manganese and copper oxides are clearly the most reactive, and we also see some reactivity of serum oxide, the silver sigma, and zinc oxide. So somewhat consistent picture in some of the cases at least. Finally, we have the DCFH2, where we have a very strong reactivity. Here we have the slope of the arbitrary dose response curve as an indicator, and we see a very strong reactivity of the carbon black. But we have several materials with interactions, so it's not really possible to make good estimations of the reactivity here. For several of the materials, including the manganese oxide, we do, however, see a reactivity of the copper oxide, which is expected. So this is clearly a problem for the DCFH2 probe in this case. So if we now try to take a look overall with the positive controls, we see a somewhat similar picture, but also variations depending on the test medium used for determination of the reactivity. For example, we saw very low reactivity in the DMEM, but significant reactivity in the serum and also in the potassium chloride when it comes to cyclic voltammetry. I would say clear picture in the sensor disk reader method with the redox potential measurements, where we do also see some similarity with the FRAS assay and the ESR in using the CPH. This, of course, needs to be further explored. We haven't digged much into this data yet, so this is coming up. But it's clear that we really need to understand the method's applicability and the achieved results. And we also need to understand the link between the results from these different test methods and their biological relevance. This needs to be addressed in future research. It's important to note that the specific medias plays a role, and there are more than one reaction pathway to obtain the oxidative or reductive behavior of the particles. So I think it's important to really think carefully about this and understand how we should test reactivity in the future. So conclusions: we determined the solubility limits and rates in all relevant cell incubation media and the key biological compartment simulant fluids. We saw important differences in the solubility and rates. We should note that the solubility also differs between cell incubation media and would result in different limits of particle dose when we do in vitro experiments. We saw the pH and redox reactivity was successfully determined in all relevant cell incubation media, and also the simulant fluids for long lining and lysosomes and standard media. We should pay attention to that reactivity tests using different test methods give different results and sometimes contradicting results, and we really need to further understand this in future research and select the most appropriate methods to determine reactivity relevant for the biological experiments. Thank you very much for your attention.